GPCR stands for G protein coupled receptor. Many medicines and hormones shows their effect via the signaling pathway. This lipid bilayer is called cell membrane. This is extracellular region and this is intracellular region. GPCR is called G protein coupled receptor because it is attached to G protein from cytosolic site. This receptor turns around cell membrane by 7 folds, due to which it is called serpentine or 7 transmembrane receptor. This end is called N or amino terminus. The cytosolic end is called C or carboxy terminus. This G protein coupled receptor form 3 loops around cell membrane. G protein is attached at the end of third loop by non-covalent interactions. G protein is a trimeric protein, that is, it is made of 3 subunits, G alpha, G beta, and G gamma. G alpha and G gamma are attached to cell membrane by 2 different polypeptide chains. G alpha is bound to GDP also. This form of G protein, when the 3 units, that is G alpha, beta, and gamma, are united together, and G alpha is bound to GDP molecule, is called inactive state. And the signaling pathway now is in inactivated phase. But when ligand comes and binds to the receptor, a transcription factor called guanine nucleotide exchange factor, abbreviated as GEF, gets activated and converts GDP into GTP. Now you can see that G alpha is bound to GTP. This GTP bound splitted G alpha subunit of G protein is called activated form of G protein. In cell membrane, an enzyme called adenylate cyclase occurs, which is responsible for cyclization of ATP molecule. After activation of G alpha subunit, adenylate cyclase act over ATP and converts it into cyclic AMP. Another heterotetrameric enzyme called protein kinase A is present in cytosolic region. It is made of 2 regulatory and 2 catalytic subunits. Cyclic AMP bind to regulatory subunit and weakens the interaction between regulatory and catalytic subunit, due to which regulatory and catalytic subunit splits apart. This splitted catalytic subunit are called activated form of protein kinase A. The former form, in which regulatory and catalytic subunits were combined together, were called inactivated protein kinase A. So now protein kinase A is activated. Kinase enzymes are responsible for phosphorylation of substrate. This activated protein kinase A goes to nucleus and phosphorylates a substrate called cyclic AMP response element binding protein, abbreviated as CREB. Now this CREB gets phosphorylated. The phosphorylated CREB will undergo transcription and will show the effect of binded ligand. Now let's do some questions. Question number 1 is, GPCR have their dash outside the cell. Amino terminus, carboxy terminus, alpha helix, beta helix. You can see in this diagram that N terminus or amino terminus is outside the cell. So answer is A. Next, on cytosolic side of GPCR, G protein bind to amino terminus, second loop, promoter region, third loop. In this diagram, you can see that G protein is bound by the receptor at the end of third loop here. So answer is D. Next, inactive conformation of GPCR is stabilized by covalent, non-covalent, hydrogen or ionic bond. Inactive conformation means when G protein is in trimeric form, that is G alpha, beta and gamma are united together. And G alpha is bound to GDP molecule also. Whenever there is reversible interaction in biological system, there is always non-covalent interaction among substances which undergo that pathway. So answer is B. Protein kinase A is dash completely inhibited by cyclic AMP. No, cyclic AMP activates protein kinase A. Allosterically activated by cyclic AMP. Allosteric activation means activation by binding to a site other than actual site of action. We know that cyclic AMP binds to regulatory subunit of protein kinase A, which weakens the interaction between regulatory and catalytic subunit, leading to splitting them apart. Regulatory and catalytic subunit splits apart. Now, this catalytic subunit, splitted catalytic subunit, is called activated form of protein kinase A. This will further carry the signaling pathway. So, actual site of action is this catalytic subunit, but the cyclic AMP molecules are binding to other site and then it is carrying on the action. So, this is called as allosteric activation by cyclic AMP. So, answer is B. Next, hormones or ligand can be considered as first, second, third, fourth messenger. Hormones or ligands carry the first part of the signaling pathway, means they activate the signaling pathway by binding to the receptor site. So they are first messenger or primary messenger. Next, cyclic AMP is first, second, third, fourth messenger. Cyclic AMP carries the second part of the signaling pathway. So it is secondary or second messenger.